We're going to have a session now dedicated to how the IHCC cohorts might be able to coalesce and explore significant scientific opportunities as it relates to the COVID-19 pandemic. We're most fortunate to have two incredibly gifted and talented experts helping us navigate this area. Kelly Althoff from Johns Hopkins Bloomberg School of Public Health and Kelly Gabo from the All of Us Research Program, who are both going to be co-chairing this next session. I'll turn it over to you guys.
Thank you, Jeff. Good morning, good afternoon, good evening to all of you. We really appreciate the opportunity to be here. On behalf of Carrie and myself, we'd like to welcome you to the first session of our dual sessions on COVID to explore if and how the IHCC should come together to assist in the current pandemic. Specifically, we'd like to know what are the COVID-19 research questions that a global collaboration of cohorts is uniquely positioned to answer. As you know, this has been a rapidly progressing situation with the numbers of infections escalating quickly. From the Johns Hopkins website, we can see what the pandemic looked like at the end of January when it was primarily in China, to mid-March, to today, when over 3.2 million persons have been diagnosed and over 233,000 have died as a result of this disease. We believe the IHCC is uniquely poised to make a difference in this pandemic and hope that by the end of tomorrow, we'll have identified working groups to tackle these important research questions. As you know, a survey was sent around prior to this meeting asking what types of data that we would be able to ascertain, what you're currently collecting, and what you might be interested in collecting as part of our efforts in COVID-19. Thank you to all of the cohorts who completed this survey. We've assembled those results and we'll be presenting them tomorrow. As a result of those surveys, we've identified several topics and have selected speakers who will provide background on these areas. In addition, we've asked each of our speakers to come up with 3 to 5 potential research questions in these areas that could be answered by the IHCC. Before I introduce our esteemed speakers, I'd like to go over a couple of ground rules which are very similar to what we just went through in our first session. We're interested in your questions and feedback, and we'll be asking the speakers to address the questions at the end of the session. Please put your questions in the Q&A like you did last time. Each speaker is going to have 10 minutes, and we would respectfully ask that they try to stay within their limit. In order to reduce the back and forth between us and the speakers, I'm going to introduce them right now, and then I'm going to ask that they go from one speaker to the next when their next title slide is showing. So on behalf of Carrie and I and the IHCC Steering Committee, we'd like to welcome our 4 guest experts. Dr. Liz Cerulli, a principal scientist at Helix, who will talk about the utilization of the Helix cohort for COVID-19 studies. Dr. Eileen Scully of Johns Hopkins, who will talk about sex differences in response to viral infection. Dr. Kerstin Koenen, professor of psychiatric epidemiology at the Harvard School of Public Health, who will talk about mental health and resilience in the COVID-19 pandemic. And Dr. Kauri Stephenson, the CEO of Decode, who will discuss his recent New England Journal article on the epidemiology of SARS-CoV-2 in the Icelandic population. After Kauri is done, Jeff is going to lead a discussion of the Wellcome Trust, and then we'll come back for more questions and answers. So I'm going to turn this over right now to Liz, and then we'll ask each of our speakers to go after that. Thank you.
All right. My name is Liz Cerulli. I'm a principal scientist at Helix, and I'll be talking about how we've been using our data at Helix to try to contribute to the COVID-19 research effort. So one of the goals that has really been clear in part of the human genetics community recently has been to identify genetic variants that cause differences between people and how they respond to SARS-CoV-2. As you heard Francis Collins say earlier, many people who become infected with SARS-CoV-2 don't show any symptoms at all, and there's a huge variety of how people respond, including all the way to people losing their lives to this disease. We already know about several factors that influence how people respond, including age and the different comorbidities that people can have, but there will also be genetic variants that influence these differences, and we do not yet know what they are, and we need to identify them. So how is Helix contributing to this effort? At Helix, we perform exome plus sequencing. This means for every person that we sequence, we produce a high coverage clinical grade exome, which lets us get at all of the coding variants that person has, including the rare and even unique coding variants that they possess. And we also have a GWAS backbone as part of that sequencing, which means that we can impute common variants genome-wide and perform any type of GWAS analyses that would be standard. We have sequenced hundreds of thousands of people with this assay, And we have a core set of a little over 200,000 unrelated individuals that we've been using for some research projects. So before we had any COVID-19 phenotypes collected for these sequenced individuals, we wanted to ask the question of what can the genetic variation that's present in our database tell us that might be of interest to and relevant to SARS-CoV-2 infection. So in particular, we started with the ACE2 gene, which is known to be the human receptor for SARS-CoV-2 and is used by SARS-CoV-2 to enter human cells. So we wanted to characterize what type of variation is present in this gene, and in particular, if there are people who are deficient for ACE2. So looking at our database of over 200,000 unrelated individuals, we identified 332 coding variants in ACE2. That are shown here, broken up by annotation and by how many people in the database were carrying that particular variant. You can see we saw a large number of missense variants, a smaller number of loss of function variants, and also a few copy number variants. I'll talk about the copy number variants really briefly. We're able to identify copy number variants as long as they impact at least one exon of a gene. And we did identify a small number of copy number variants in individuals in this gene. The most common one was duplication of the entire gene, which occurs in 0.04% of people. Any of these variants will be of interest to study with regard to COVID-19 phenotypes, but the most interesting ones will be the ones that are in the regions of ACE2 that bind to SARS-CoV-2. So shown here is a lollyplot of ACE2, and the regions of ACE2 that bind to SARS-CoV-2 are highlighted in those black boxes. Within each black box, you also see a number of little red ticks, and those are the specific amino acids that have been shown to interact with SARS-CoV-2. Each point in the lollyplot is a genetic variant that we identified in our database. The height of the stick indicates the allele frequency. The number within each circle indicates how many people in the database had that variant. And then these are all missense variants, and they're colored by how damaging they're predicted to be, with the most damaging ones shown in darker colors. All in all, we found 40 different coding variants in the regions of this gene that bind SARS-CoV-2, and that corresponded to about 1% of people. If you look just at the very specific amino acid positions that interact with SARS-CoV-2, we identified 11 variants at those sites that would change those amino acids, and that corresponded to 0.04% of people. These variants will be of extreme interest to study because any of them that block the binding of ACE2 to SARS-CoV-2 could produce immunity to infection, and any that produce enhanced binding could make people more susceptible. The other thing that we looked at with ACE2 was what happens with loss of function of this gene. So there are considerations when trying to design therapeutics to help prevent SARS-CoV-2 infection for whether or not loss of the ACE2 gene is a viable option for humans. So it was already known from public databases like gnomAD and the PLI score that loss of function of this gene is not well tolerated. But since this gene is on the X chromosome, we wanted to take one additional look and see if the frequency of loss of function variants was different between males and females. Because males only have one copy of the X chromosome, loss of function variant, if deleterious, would be expected to be much more depleted in males than in females. So shown here is another lollyplot of this gene, and once again, missense variants are shown in blue. Now the entire gene is being shown, and the loss of function variants are shown in yellow for splice site variants and pink for stop gain and frame shift variants. And you can the females are plotted up top, and the males are plotted on the bottom. And you can just visually see that there are fewer loss of function variants in the males than in the females. And indeed, when you add all this up and put together the total loss of function allele frequency in males and females, you see that the allele frequency of loss of function variants is much lower in males than what would be expected given females, and this is a statistically significant difference. So loss of function of this gene is not well tolerated. So that's what we did with our database before we had COVID-19 phenotypes, and we put out a preprint on bioRxiv about a month ago now, making those data available. But of course, what we were most interested in doing was collecting COVID-19 phenotypic information. So that's what we embarked on next. A large proportion of the people whose exomes we sequence consent to participate in research studies, and we're able to recontact them after the fact to gather additional phenotypes from them. So what we've been doing is we've been sending out surveys, COVID-19 phenotyping surveys, to our research participants, and some of our partners have been doing that as well. We've now received survey responses from about 25,000 research consented individuals. And I'll just quickly walk through some of the stats from that. So we have 17% of people who report that they've been exposed to someone who has a COVID-19 diagnosis. We have 12% of individuals who experienced a fever during the pandemic, and 6% of people report kind of those classic COVID-19 symptoms of having a fever, dry cough, and a loss of sense of taste or smell. Out of the respondents, 3% have actually taken a COVID-19 test, and 10% of those test results have been positive. Because all of these people were already sequenced with our Exome Plus assay, we already could jump right into doing genetic analyses as soon as the phenotypes came in, which was it was about a week and a half ago that we started getting the phenotypes in. So this is all very, very fresh data. So right away, we were able to do common variant GWAS. We were able to do rare variant analyses, including the rare variant gene-based collapsing analysis that's demonstrated in the top right graph here. We also were able to look at CNVs and HLA types and look for associations with all these different types of genetic variation. We're doing this genome-wide and also on with a more of a focus on candidate genes like ACE2. And we're able to do this with a number of different phenotypes because the surveys we've been sending out are pretty comprehensive. They collect lots of information about different symptoms that people have experienced, different exposures they've gone through, different medications, comorbidities. We're trying to be really comprehensive and to global standards when collecting these information from people. So far, our results are, of course, still pretty preliminary because the data are pretty fresh, but it does look like there's not going to be a simple, easy genetic solution to the question of what variation is making people have different responses. There have not been clear signals that have jumped out yet, although, of course, there's still more analyses to do, and we're not done yet. So to the effort of trying to get this sample size up so that we can make actual discoveries. We are also part of multiple different worldwide consortia that are trying to pull together COVID-19 host genetic information. Two of these efforts are the COVID Human Genetic Effort and the COVID-19 Host Genetics Initiative. We actually made our GWAS results available to the COVID-19 Host Genetics Initiative already, and they just completed their first meta-analysis across multiple groups late last week. They just released their results, I think, on Thursday or Friday of last week, which is the Manhattan plot shown in the bottom right. This is from a meta-analysis GWAS that included 917 positive COVID-19 cases versus population controls. There were no genome-wide significant hits at this point, so this is clearly something where there's going to need to be a larger sample size and more finesse on the phenotypes that people are looking at. But it's been really great to be part of these global efforts because it's wonderful to see scientists coming together to really try to make these discoveries. Not only are we all sharing the results of our analyses with each other, but we've also been working together to build the types of surveys that we're administering, the protocols that we're using, and the ideas for analysis. So it's making it so that we can really harmonize the types of analyses that we're doing across these groups. And I feel like that's really in line with what is happening with IHCC as well, and that will be a great contribution to this effort. And I'm excited to see how getting all these data together can really help. And the next slide is just my acknowledgements. Many people contributed to this work at Helix. Thank you.
So Liz, this is Eric Plummer. We do have a question Alicia Martin has posted. The question is, since the X chromosome has a different effective sample size than the autosomes and is subject to different selective pressures, are the male-female differences in loss of function frequencies in ACE2 greater than expected relative to X versus autosome generally?
Do you mean, I assume you mean, did we account for the fact that females have two X chromosomes and males have one? And yes, it was about the allele frequency in each group, not like the proportion of people who are carrying a variant. It was based on the allele frequency.
Okay. And Terry, did you have a question as well?
Oh, I did. Yeah, I just asked, if any of the male-female differences were significant in the frequencies, and it sounds like some of them were from the loss of function, but on X chromosome in general.
That's right. Yeah, we were looking to see if any other variants were showing depletion in males the same way that loss of function variants did, and no, we didn't see signals for the other variants. It was just the loss of function category at this point.
Great, thanks.
Okay, so I think the idea from Kelly was for us to just keep rolling through. Could I have the next slide, please? Thank you. So my name's Eileen, and I'm going to talk about sex differences in responses to viral infections and how that might intersect directly with the COVID-19 pandemic. So for the purposes of this discussion, sex is considered to be defined at birth, determined usually by chromosomes and anatomy with a superimposed influence of hormones, whereas gender reflects more the socio-behavioral context and a lived gender identity, and includes also environmental exposures and also potentially exposure to hormones. And so focusing our discussion here on the realm of infectious disease, I think there's a classical understanding of this as a monolith of sort of the person versus the pathogen, with some granularity added by considering the life course. But I think it's important to remember that there are sex-specific immunologic demands, and most notably the need to tolerate a pregnancy, and then the need to transfer protective immunity in the neonatal period. And this leads to fundamental shifts in how the immune system needs to function that leave an imprint on the immune system response to pathogens. And I mention this just to frame the discussion, because I think otherwise it's somewhat surprising to think that there would be sex specificity to a response to pathogens. So sex is known to be a modifier of the response to viral infections, and this is notable for 2 at least chronic viral infections. So hepatitis C, where females have an odds ratio of greater than 2 for spontaneous clearance of disease, and in HIV, where there's a clear higher probability of achieving spontaneous viral control, which is a rare outcome but does occur among women as compared to men. In considering something like other pandemic infections like influenza, there are sex differences in the severity, and this varies over the course of a lifetime, with males generally having more severe disease in the extremes of age, but during the reproductive years, females have an excess of mortality. So thinking specifically about coronaviruses, what can we learn from prior episodes? So the severe acute respiratory syndrome, or SARS epidemic of 2003, and the Middle East respiratory syndrome, MERS epidemic in 2013 to 2014, were both caused by coronaviruses and had higher mortality reported among males. It's important to note that there's probably some exposure variables here, in particular for MERS, where it was known that being a camel trader was a higher risk for acquiring infection. However, in a murine model of SARS-CoV-1, male sex was again associated with higher mortality and linked to estrogen exposure in that model system, suggesting that not only gender but also sex is a modifier of these coronavirus infections. So what about in COVID-19? So one of the early signals from China was that there was an increase in risk of death among males. And so these data are just beginning to be published, but there is a higher mortality rate for males in at least one of the public data sets available from China, and more severe disease in case series from individual hospitals. In addition, there was a higher risk among those who were severely ill, higher risk of death among males who were severely ill as compared to females, and then a suggestion that there was a greater added risk associated with comorbid disease that was observed among men. And in the Global Health 50/50 website, which has been tracking COVID-19 and using all of the available data that is disaggregated by sex, summarized data from 77 countries with either full or partial disaggregation, confirms that there was a difference in the male to female ratio of deaths, whereas a similar difference was not really observed in the number of cases. Next slide, please. So what are the remaining questions? So I think one of the biggest remaining questions is what the role of asymptomatic infection is. So in about 60% of the cases that were identified in the Republic of South Korea were among women. And it's really notable that in South Korea, the screening strategy was not based on symptoms or severe symptoms, but was much more widespread. In addition, if you look at data from China, in a recently published paper in the Lancet Infectious Disease, they used 2 different methods of identifying infected individuals in one of their case tracing. One was symptom-based surveillance, for which 55% of the cases identified were men, and one was contact-based surveillance, and in that case, only 28% of the cases identified were among males. So this does suggest that there may be an increased frequency of asymptomatic or minimally symptomatic infection among women, which would mean that the case fatality ratios are actually underestimated, and that there's an even more substantial risk of male sex that's associated with poor outcomes. In addition, there are also other behavioral factors and other things that need to be considered in this data. So the risk of acquisition may also vary. In one of the outbreaks in the early United States experience, about 68% of the residents of a nursing home who became infected and 76% of the staff in a nursing home who became infected were female. However, we don't actually have the denominators for those numbers, so we don't know the proportion of residents who are female and the proportion of staff. And I'll just note that of healthcare workers at risk in the United States, about 75% of them are female. So again, considering the role of sex and gender is going to require really careful analysis of the data and a real determination of what the denominator is that we're considering. The other thing that needs to be addressed is the role of sex over the life course. And so this is disaggregated data by age and sex. And in some of the data sets that are available, they provide both age and sex disaggregation. And so what we can see here from Italy on the left here shown is the cases per 100,000. And on the right is the deaths per 100,000. And you see some difference in the rate of identification of infection and then this substantial difference in death rates. And those two things need to be intercalated and also considered in terms of the population that has lived to some of these ages for which we also would expect to see a sex difference. Could I have the next slide? So just to summarize, I think the data so far suggests that women are efficiently infected with SARS-CoV-2. They also suggest that COVID-19 severe disease and mortality are more common among males, and that this mortality risk difference persists into old age. And that last point there, I think, really underlines that while sex hormones may be a driver of many differences in immune responses, it's unlikely that that's the only difference that's operative here among females. And so just as sort of a framing for this group, I'm a basic science and translational researcher who usually looks to the kind of research you do to validate that anything we're doing has any actual importance. But I think there's a couple points where we can consider how sex and gender may intersect with this disease. So this could be at the risk of acquisition and receptor expression for viral entry in the upper respiratory tract or in the early innate immune responses to that new exposure to infection. It could be at the progression to lower respiratory tract infection, again, where receptor expression may play a role, where the early innate responses may play a role. But here we're getting more into the adaptive immunity, the recruitment of cells from other locations, and the antibody responses, and then potentially intersections with comorbid diseases, including smoking, COPD, and obesity. And then finally, we have this progression to severe disease where we have severe alveolar inflammation and leak. And again, at this point, we need to see the repair processes, the clearance of infected cells, and importantly, the balance of inflammation and resolution of infection, which may also have sex specificity. So thinking about this on a cohort level, which I don't usually do, so please excuse the naivete of some of this. So I think from a genetics perspective, there's the genetics of the viruses, and then there's also the genetics of the host, as like beautifully introduced by the talk right before me, and understanding how that may intersect with either receptor expression to establish infection or in immune variations in the response that may be either protective or detrimental. Next slide, please. And superimposed upon that are the effective sex hormones. And there are direct interactions between hormone exposure and genetic effects, but also hormones have an influence on the development and mounting of protective immune responses, as shown by the variation in susceptibility to infection through the reproductive years in women. And finally, next slide. Added onto those things are the bidirectional interactions between comorbidities. There are genetics that put you at risk for comorbidities, and there may be also comorbid conditions, including diabetes or COPD, that could make a genetic risk allele more important in the outcome of infection. And these also have bidirectional interactions with hormones and other sex and gender factors. So I think thinking about cohort level questions, I think there's a real importance of defining the populations at risk and the outcomes at each stage of disease. I think on top of those things, understanding the cohort level contributions of genetics, comorbidities, and hormones is really critical. And then putting that into the context of the variations over a life course. I didn't really talk about it at all, but I think it's been well described that there is a minimal risk for severe infection among children. And understanding how that risk is modified is also another key question. So just to summarize a few thoughts about how these things may be used to uncover pathogenesis of SARS-CoV-2 infection. I think the integration of age and sex into identification of immunologic features that are protective against severe COVID-19 will be really useful to understand pathogenesis. I also think that there are likely to be sex-specific genetic features that influence the severity of disease, and that in order to identify those, there's going to have to be very careful partitioning of the types of disease manifestations that are observed, not just acquisition of infection, but whether or not you progress to severe disease. I think there's also a lot of work to be done in the intersections between sex and comorbid diseases, and really critical to understand the true denominator of the pandemic, to understand variations in transmission and in the heterogeneity of disease. And I think those are all of my slides, and I can either take questions now or at the end.
So I saw there was one question about, is the higher male mortality risk adjusted for pre-existing conditions?
So the short answer to that is no. In the early emerging data that's coming out from China, there's only been 2 peer-reviewed publications that have looked at this now, but there are numerous in various stages of preprint and review. It looked like comorbid disease in China had a bigger impact on outcomes in men. Unclear which of those comorbid diseases they were referring to. Early reports also cite the much higher levels of smoking among men in China.
Perfect. And then there's one question about behavioral factors and other lifestyle factors and the opportunity for exposure. And could that be part of what is causing the increase in mortality in men?
I think this is a huge question, but I would suggest that, with the risk of acquisition, in particular in places like in the United States, where 75% of healthcare workers at risk are likely to be women, we really need to understand the prevalence of exposure and infection as opposed to symptomatically defined infection, which has been sort of the high level. So, for example, in New York City, testing was restricted to people with fairly significant symptoms or comorbidities. We have no idea the true prevalence of infection, and the data suggests that actually females are more likely to be infected and to be asymptomatic. So we need to understand those differences. And I think cohort-level studies of seroprevalence would be a critical aspect of defining that.
Terrific. Thank you, Eileen. All right, maybe we'll move to the next speaker. Thank you, Eileen. Carson?
So I was asked to talk about mental health and resilience in the COVID-19 pandemic. And although I do psychiatric genetics, this talk will have no genetics. So I'm going to talk about COVID-19 from a little different perspective than any of the other speakers, and why it's in terms of why it's important for us to consider mental health. And perhaps this will be a motivation for people to be looking at this or considering this in their cohort studies. Because I do think cohort studies have a really unique opportunity to study mental health here, because we actually have pre-exposure information or pre-exposure to COVID-19 information. So we've all seen the images. These are ones, some of these are local for me in terms of the multiple stressors we're experiencing in addition to the risk of being infected by COVID-19 with park closures, shortages in some of our stores, some places, New York, Italy, bodies piling up, the empty streets in Times Square. And so COVID-19 has a bunch of characteristics that we know from decades of research, are toxic to mental health. It's novel, it's unpredictable, it has a high level of threat, and people feel out of control. On top of that, we're experiencing social isolation, elevated rates of bereavement, and in certain communities, stigma, which are all in themselves toxic to mental health. So it's really a perfect storm. We also know from other epidemics that there's mental health consequences. So, for example, there has been studies during the SARS epidemic about increased rates of suicide among older adults in Hong Kong, particularly related to social isolation. And even following up SARS survivors many years later, they found that they had higher rates of psychiatric and other consequences. So even though they had recovered from SARS, there were these persistent neuropsychiatric consequences of SARS. In terms of Ebola, studying people in the community a year later, they found that there were high rates of depression. You can see here depression, PTSD, and anxiety. And they were especially high in people with Ebola-related experiences, that with people in communities that had deaths from Ebola or were exposed to a lot of Ebola. Then we have data coming out from China that suggests there's wide-ranging adverse effects on mental health. So this is particularly among healthcare workers, and also in the community, among people who were healthy, actually, so not sick from COVID, but even just exposure. We saw this after 9/11 terrorist attacks in the US too. Exposure to the sort of media and concern around COVID, even if you weren't directly affected, was related to higher rates of depression and anxiety and other psychiatric symptoms. And so this, you can see the prevalence of they're talking about is about, half the population, just showing that many people normally who would not have mental health problems are just showing some significant symptoms. And this had come out recently that colleagues in China are talking about a public mental health crisis. So what can be done? And I'll talk about a few things we're doing and other people are doing. Next slide, please. So I think we need to consciously from a public health point of view, address the mental health in the COVID-19 pandemic. And it's clear that first we need to address contain the disease, the virus. But we do have the potential to sort of get ahead of some of the mental health problems that we've seen in China and that are emerging in other places. So just like we have a do the 5, this is from Google or other sort of catchy phrases on what we can do to prevent the infection from COVID or prevent the spread. We can do the same in mental health. So some colleagues I work with have proposed a 5 for mental health. And the first thing is recognizing that there is a potential for a large public mental health problem. So recent data, national polling data, so I showed the data from China, but national polling data in the US also suggests Americans are worried about their mental health. And in other places too, but this is just the data that came out most recently that I'm aware of. So for example, this is from Kaiser, although there was more recent, something came out more recently this week after I sent my slides in, that a majority of 47% of adults are saying that sheltering in place is having an adverse impact on their mental health. And other groups as well, people with children, et cetera. So Americans are saying that this is affecting their mental health. And again, it's true in other places as well. Those are just the data that came out when I was doing the slides. And so what can we do? So one thing is expanding the safety net. So we know that the other thing that in terms of mental health that we're concerned about is the economic fallout itself will have adverse effects on mental health. We and other people published data from the 2008 Great Recession that showed that people who had home foreclosures and lost their jobs had increased incident depression and anxiety. So the combination of COVID-19, the virus, the social distancing measures, which are critical, and the economic fallout are really challenging for mental health. And one of the first things we can do is support these social safety net practices, because if we can keep people in their jobs and their homes and keep their income, that actually is protective for mental health and actually for their health broadly. So this is just showing that we showed that home foreclosure during the financial crisis of 2008 was associated with increased incident depression and anxiety. Also, there's a lot of efforts aimed at assisting those most at risk. So those include a lot of populations, a lot of groups, which is an argument for a more population approach to this. So there's people with existing mental health problems, people suffering from addiction. There's been reports of increased rates of domestic violence or child abuse because families are stuck together, and also the normal systems, sort of schools, et cetera, that monitor these things are closed. Families with special needs children, anyone already socially isolated, and healthcare workers, et cetera. So one of the things people can do is find a way to help in their community. This is just pictures from my own neighborhood where people are making masks and delivered them to hospitals earlier this month, but also doing science and studying mental health in our cohort. So just a couple of quick examples. So one is in the nurses' health studies in the Harvard cohorts. We have done a initial cohort survey in addition to collaborating with Andy Chan and Tim Spector on the Zoe app to monitor COVID. So in the cohorts, we invited the cohort members to join the app, as well as we're doing baseline surveys that include many of the things, including identifying people with COVID-19 and who've been tested, but also studying mental health, and probably as important, studying protective factors, so that over time we can look at, in the broader cohorts, mental health effects of this crisis, but also what protected people, and then also link this to our broader cohort data, which will allow us to look at other diseases down the line. I have colleagues at Boston Medical Center and elsewhere who are saying some of the other consequences of COVID, the cardiovascular and the neuropsychiatric consequences, are being underreported. We've also been working with people who have pregnancy cohorts to incorporate mental health, because so many of the women are talking about mental health, emergence of mental health problems related to their concerns about COVID, the stress of things like worrying about giving birth in the hospital by themselves without their partners, about worrying about their babies being infected, et cetera. And then finally, I can't talk about mental health without talking about cultivating resilience. So many people, one of the positive things that may come out of all this is that there is so much online about mental health and resilience and coping. We've been doing these mental health forums, if anyone wants to join, where we've been kind of disseminating evidence-based practices on how to protect your own mental health during this time. And then finally, I think one of the things that people always ask me when I talk to media these days is what is one thing we can all do during this time to help to protect our mental health? And that is have empathy for ourselves and each other. So these are just pictures from my neighborhood, which showed ways when I was walking my dog, actually. So people are putting signs out for people who are delivering, and essential workers who are still working. And then I found that someone in my neighborhood was leaving these rocks with these words on them. So when I was waiting for my dog to go to the bathroom, I saw this, and it sort of made my day. And I just thought it was a really empathic, a very sort of small empathic thing that people were doing in my community. And I think that there is a science behind empathy and altruism, and that by doing things that are good for other people, it actually kind of biologically makes us feel better. And so I'll close with that. Thank you very much.
Great job. Thank you. I see a number of comments regarding collecting additional data, and we'll get to that a little bit tomorrow.
I think there was one question with regards to and I think this is going to maybe be for all the speakers in this session, is how are specific ways that the IACC, the cohorts, can address the issues that we're discussing.
And I can put it in the chat. I mean, we've been sharing, I think people who are doing mental health and economic data are all sharing, and people are sharing about that. So they have questions. I can just put that information in the chat from my end.
Terrific. Thank you. All right. Maybe we'll transition to our last speaker, certainly not least, Kari Stephensen.
Hello. And I'm just going to give you a little overview of our study of COVID-19 in Iceland. Next slide. The first case, or screening for the virus, began in Iceland in January 31st. And the healthcare system was screening people coming from regions that were considered high risk. And they had been screening for a whole month before finding the first case on February 28th. And it was then on March 5th, when I was driving to work, listening to news on the radio of the epidemic in China, and they were talking about death rate. And if I remember correctly, they were assuming it would be about 3.4%. And I couldn't understand how they could calculate our death rate without the denominator, without knowing the distribution of the virus in society. So we decided to screen the general population in Iceland. The healthcare system was screening people with signs and symptoms and people coming from high-risk areas. We started to screen the population in general. And yesterday we had screened 32,000 Icelanders, and the infection rate was 0.58%. And yesterday we had screened 32,000 volunteers, and yesterday we had also screened about 4,000 randomly selected individuals, and their infection rate was about 0.54%. The healthcare system yesterday had screened about 18,000 individuals, and of those 9.6% were positive. So yesterday we had screened 14% of the population of Iceland, which is a remarkably high percentage. And actually, only about 12 days after we started the screening, we had submitted a manuscript to the New England. And most of what I'm talking about today is at least covered in some way or another in this paper. If you go to the next slide. In addition to screening the population, screening volunteers, screening randomly selected individuals, and screening those at high risk and with signs and symptoms, we have sequenced the virus from every individual infected in Iceland. And there is a remarkable number of mutations. We find about 1,100 mutations in the virus, distributed basically all over the genome. And I want to emphasize that there are even mutations in the receptor binding domain of the spike protein. And actually, when you begin to look at the sequence of the virus, that even though it has a relatively low mutation rate, it has infected so many that there are this incredible number of mutations found. And actually, when the virus moves into a region and continues to mutate, mostly randomly, there are haplotypes that arise that become fairly specific for the region. There is, for example, a haplotype that is fairly specific for Austria, another for Italy, a third for England, a fourth for the west coast of the United States. But the European haplotypes are basically the A to A haplotypes. But all of this, of course, comes out of China. And what is interesting, since we have sequenced the virus in every single individual who has been infected in Iceland, we can use this haplotype structure to determine the geographic origin of the infection in every single individual. And we can also use it to track the infection in our society. So what you're looking at here is the picture with sort of clusters of infections, and the colors indicate the haplotypes. And towards the bottom, there is a big blue cluster which consists of members of a church choir, who all of them got infected by the same person coming from a ski vacation in Italy. And actually, you can use this easily to track infection. And this picture is simply showing you one example of where the T25 is an individual who is heteroplasmic for a mutation. He has the virus with and without a particular mutation, but showing that everyone who got infected from that person contains the mutation, which is probably due to drift, but it raises the possibility of higher infectivity of the mutated virus. What is interesting about this, as the epidemic progressed in Iceland, there was a change in the haplotype configuration in those infected. In the beginning, they were mostly from Austria and Italy with the A to A haplotype. But subsequently, the A1 haplotype, which is specific for England, began to emerge. So as the authorities were focusing on people coming from ski vacation in the Alps, the virus was sneaking into the country with people coming from other countries, such as England. So it is Great Britain. So it is clear that the virus had much wider distribution in Great Britain very early on than people realized. And I'm just going to show you for fun of it. This is showing how the haplotype structure has changed in the United States. I mean, in the beginning, it was the B1A haplotype on the west coast in California and Washington. And then gradually, the A to A2 haplotype has taken over. And then the big question is, why did that haplotype take over? If you go to the next slide, it was not because of any kind of a selection, because what you see here is the plotting of cases in various parts of the United States as a function of number of individuals who arrived from New York in that particular place. So it simply seems to be because of the way in which the Americans are moving around and carrying with them, in this instance, a particular haplotype. What is interesting is that early on in the epidemic in Iceland, most of the cases were transmitted from people coming from abroad who had been traveling. Then it became sort of social interaction, but of late it has been mostly due to transmissions within families. And that basically shows you that the measures that the authorities took in Iceland are working. And what were these measures? It was to screen widely, as we've done. We have screened 14% of the population. We have an extraordinarily agile team tracking the contacts of the infected, putting the infected into isolation, and putting the contacts of the infected in quarantine. And actually, we kept people in isolation for 2 weeks, and then insisted that they had to be without signs and symptoms for 1 week before they were considered cured. And this is basically a plot of the transmission rate from the 13th of March to the 1st of April. And during that period, it declined by about 2% a day. This is basically from a screenshot from the website of the authorities from today. Altogether, we have had 17 199 infected, and we had the whole genome sequence of all of them and all of their health history from birth. There are currently only 4 individuals in a hospital. There is no one in an intensive care unit. We have had about 6% of the nation in quarantine. Now we have only 577, and we have screened over 50,000 individuals. And go to the next slide. This is just showing how this current phase of the epidemic is fading out in Iceland, where active cases are very few. The number of individuals towards is very large. And go to the next slide. I want to emphasize that we have done a fairly thorough search for association of variants in the sequence of these 1,800 individuals with and the control population. We haven't found any association. There is no association with ACE2 in this population, at least. There is no association with anything in the HLA region. We have, in Iceland, it's absolutely clear that not only do women not get as sick as men if they get infected, they have clearly less tendency to become infected, and this is based on an entire nation. Children have less tendency to get infected, and they do not become as sick as the adults. We have recently started to do antibody screen. We have screened now about 6,000 individuals, and there are interesting patterns coming out of it. We have joined the international effort, consortia, to look for the host genetics, and we are also very much focused on looking at the potential associations between sequence diversity in the virus and the clinical outcome of those infected. And we are participating in various efforts to develop therapeutic antibodies. And that was basically it.
We have a question from Philip related to potential mutation rates. Does DECODE capture virulence or titrate for individuals?
We haven't found any association between the mutations and clinical outcome. We have been focusing on it. You see, in our case, we have only 1,800 cases in the country. We have complete data on all of those 1,800. And this is one of the instances where what you need is definitely something you don't want. We would need more cases to be able to make sense of it. But we are now in all kinds of international collaboration on this. So, we have mapped out the mutation rate. We have mapped out how it mutates when it moves from one individual to another. We have also been looking at many samples taken from the same individual to see how it is mutating within the individual. So we have the mutations very nicely mapped out, but unfortunately we haven't found any association between the rate or the pattern and a clinical picture.
Okay, and next question is from Caterina Ferraccio. When you say 50% of cured positive for the virus, have you studied if the virus is viable?
50%, what did you say?
The question is, when you say 50% of the cured tested positive for the virus, have you studied if the virus is viable?
No. Unfortunately, we don't. But basically, after 2 weeks of isolation, 1 week without signs and symptoms, half of them test positive for the virus. There's no question about it. Their CT value is increased from the time they are diagnosed almost always.
Carrie, this is Jeff Ginsburg. I had a question about whether you think the rate of change in variants in this virus is similar to or very different from other circulating viruses like influenza. And based on your data today, would you predict or speculate whether vaccines that are being developed against early forms of the virus will be effective or potentially not effective a year from now?
Oh, you see, this is one of the big questions. This virus does not have a particularly high mutation rate. It has, however, infected so many that it has had an ample opportunity to mutate. And there are certainly there are mutations found in basically most of the residues, not in the one that makes the alpha helix in the spike protein. That seems to be invariant. But this is one of the things that we will have great difficulties predicting. I think that we should be optimistic that vaccines will work, at least in the short term. But we should certainly not let it take us by surprise if we will have to continue to develop new vaccines into the future.
Next question is, given that you saw heteroplasmy, is this extensive, and is any of the viral sequence diversity driven by recombination?
I am not sure that any of it is driven by recombination.
This is Terry Manolio. I had asked a question in the chat because the Q&A don't seem to work. I was wondering, Kara, if you could tell us what the IHCC cohorts would need to provide to be able to do viral mutation testing. Is it just from the simple pharyngeal swabs or nasal swabs that are done?
Yes. We just we have just sequenced this from the nasal swabs.
Great.
And it works very well. There is of course in the public database there's an enormous number of mutations coming from all over the place, and we are continuing to sequence. We have been continuing to sequence the new cases, and we are currently very much focused on understanding sort of the mechanism of mutation. We have some little bit of insight into that we are working on. But the disappointing aspect is that we have yet to find any correlation between mutational pattern and clinical outcome. But what I'm describing is basically a cohort of a whole nation, right? Where we have every single case, we have the host genome sequenced, whole genome sequenced of everyone, and we have the virus sequence from everyone, and we have very extensive clinical data. And I think that it is important for the rest of this team to realize that we should probably add this on the top of everything else that is out there, so we can figure out what this beast is really doing.
Mary De Silva here. I also posted a question in the chat. Can you tell us more about the antibody screening results and whether you used an ELISA or a lateral flow assay?
We are using an ELISA, and we have screened now, as I said, about 6,000 individuals. I want to wait until we get a second ELISA. You see, we have just done this with one ELISA, and I am reluctant to express myself on any details of it, because we took about 1,000 sera from 2017, and in this assay, 1% of them is positive. So my guess is that anyone who has a high titer of antibodies against one of these coronaviruses is going to cross-react with this. So I would really, we expect to get another ELISA this week, and it only takes us, we can screen, 2,000-3,000 samples a day, so we will rescreen everything that we have done. There is a difference in titer depending on the severity of the disease. There is a difference depending on the age of the individual. There is a difference between the sex of the individual. There is a lot of diversity in the response, in the humoral antibody response to this virus. But before I comment on details, I would love to, or let's put it this way, I'm reluctant to comment on the detail until we have gotten the second assay.
Thank you. And can I ask if you're also looking at asymptomatic positives?
We are looking at everyone in Iceland who has been diagnosed with the disease.
That's amazing. Right? Thank you.
And we are, we have screened, as I said, more widely than anyone has done. So my guess is that we are closer to capturing everyone than anyone else. So this is as complete of a picture as you can get.
Yeah, because I'm worried about the validity of the ELISAs for asymptomatic people, but you'll be able to help test that.
Repeat what you said.
The validity of the ICEs, the sensitivity and specificity of the ELISA on asymptomatic people, I don't think has been tested well enough, because most of the validation has been on people with clinical symptoms who've then had a test. But your data is different. You can actually look at that because you screened everyone.
And it's absolutely clear that the titer is lower in those who are asymptomatic than the ones who become really sick.
Thank you.
Okay, we have another question. Have you seen the same type of variation in Iceland, for example, clinical severity, as other countries?
Yes, we have seen enormous difference. There are people who describe this as a mild cold, and there are those who end up in the ICU on a respirator. And actually, the clinical severity difference between males and females is dramatic. At one time, there were 10 individuals on respirators in Iceland. 9 of them were males, 1 female. And it is clear in our data that not only do women not get as sick as men, they are less likely to get infected. Okay. And then, of course, what is interesting is that not only are children less likely to get infected than adults, if they get infected, they are less likely to get seriously ill. But what's most interesting is that children are very unlikely to infect adults. We can show that, in all of the cases we have, there's only one instance where a child infected a parent, but incredibly large number of instances where parents infected a child. And we have been able to confirm this by using the haplotypes and mutations on the background of the haplotypes. So it's fairly interesting, but it's relevant when it comes to the question, or should we keep the elementary school open? Should we keep the childcare centers open? And these data certainly support the wisdom of that.
Okay. Next question then. Can you comment on what the IHCC cohorts would need to collect to provide viral samples for mutation testing? Is it simply from the basic nasal swabs?
I already answered that question.
Oh, I'm sorry.
Yeah.
And the next question then is a person who's recovered from one hepatitis vulnerable to infection from another?
I mean, that's the same question as the question about the vaccine. We have no data in our hands that indicates that the haplotypes are distinct when it comes to the way in which it is handled by the immune system. Like we have no evidence to support the notion that they cause diseases of different severity or different in any form, or shape. We are still uncertain about all of this. And at least, let's put it this way, a very large percentage of those who have gotten infected have raised antibody against the virus.
Okay, I think we have 2 more. Have you compared virus testing with antibody testing results to see the consistency between the 2 tests?
Of course we have, because we only classify those as having been infected that are positive out to the PCR test. So we have certainly compared that, yes.
Thanks. And the last question, are infection rates estimates corrected for exposure? So looking at clusters within families.
Repeat the question.
Harry, that was your question?
It was. So when you were saying that children and women are exposed less, are infected less, is there a way of correcting for whether they were exposed?
Yes. We are looking at this within families.
Yes, that was my question. Great.
Yes.
Thanks.
Okay, I think we're at the end of our time.
That was terrific. Thank you.
Right. So this is Jeff Ginsburg. I'm going to jump in here to talk briefly about efforts going on with Wellcome. And this is related to the questionnaire dialogue that's been going on in chat and during this session. So about 10 days ago, if I'm correct, the timing, Wellcome Trust initiated a process to develop a standardized set of questions for low and middle income countries and COVID. These would be used, or at least ostensibly proposed to be used, by the Wellcome Trust funded low and middle income country cohorts, as well as IHCC low and middle income country cohorts. And about 20 cohorts are now involved in this effort as of about a week ago. The questions cover a number of domains, ranging from health, behavioral behaviors and behavioral health, mental health, economic, social, environment, and probably others that I'm not recalling off the top of my head. But the deliverable is a core set of standardized questions that will be arrived at, I believe, by May 14th. These will be hosted in REDCap and accessible to the IACC and all the Wellcome Trust cohorts. The IACC is coordinating this effort and aims to use the questionnaires, if desired, more broadly in its COVID-related efforts, as well as facilitating cross-cohort analysis, particularly among the LMIC initiative with the Wellcome Trust. And from what I've been seeing in the chat and a lot of the discussions today, that we should probably be thinking about creating a repository of relevant questionnaires for selected use that the IACC could host and provide access to all of the cohorts. Mary De Silva, I just want to make sure that I covered at least close to what the intent is for this effort that is moving very fast, and just invite you to make any additional comments.
Sure, thank you, Jeff. Yeah, no, that's exactly right. Just to say, this started with us working with a UK set of cohorts, I think about 16 cohorts in the UK, and we developed a core questionnaire for the UK, and then are now moving out to low middle income countries. And there's absolutely no reason why that also couldn't be done for other high income countries as well, and we can make the UK questionnaire available to IHCC members as well. Just again, from a personal note, it's been quite incredible how collaborative the cohort PIs have been, both in the UK and of low middle income countries. And it's just been a joy to work with all of these people to very rapidly, within a couple of weeks, across a large number of cohorts, come through an agreement for a core questionnaire, and to see that questionnaire already being rolled out to tens of thousands of people. So thank you everyone who's been involved in those efforts, and we look forward to finalising the process. And thank you to IHCC for taking on the running of it all.
Great.
Next slide. We just want to wrap up very quickly. What we're hoping to talk about in the next 2 days is how to leverage existing data and potentially thinking about collecting new data. How can the IHCC cohorts uniquely contribute to this COVID-19 pandemic? And on the next slide, we've identified some potential research topics that have been suggested by many of the people who are on this call, as well as others throughout the consortium. The idea of serology is a very important one, and looking at biospecimens that were collected from November of 2019 through the present, and then sort of moving forward to look at antibody testing, I think is a very interesting one. The impact of host genome and environmental exposures on COVID-19 and the host response, both thinking in terms of sex differences as well as other types of environmental or chemicals such as tobacco, pollution, population density. And then third, I think based on some of the really interesting information that we heard, what are the impacts of social distancing on mental health and anxiety, and what are the outcomes within this current pandemic? We are interested in your topics of interest. If there's things that you think we're missing, we're hoping that you will put them in the chat or will email one of us. We are putting together the results of the survey, which we will show tomorrow, as well as the research questions that have been proposed by the speakers and some of you who have sent them to us ahead of time. What we're hoping to do as an end result of tomorrow is to put together working groups thinking about some of these specific topics and questions, and certainly are open to other topics if there's ones that haven't been mentioned either here or throughout the discussions. So I want to thank the speakers for a terrific job in summarizing some amazing data that has really been put together in a very short time period. I want to thank the IHCC for welcoming Carrie and I to come and speak with you, and I look forward to tomorrow's discussion. So thank you for your time, and please continue to put the chat. We're following it and really excited about all the information that's being put into there. Thank you.
Thank you. I just want to wrap up this wonderful first 3 hours or so of our virtual meeting. First, to just reemphasize that during the next break, we're really going to be looking for your research ideas to go into the chat, or even after this, because we're going to be assembling them and using them for the session tomorrow to really begin to articulate our research strategy. So just to recap very briefly, what we did this morning. First, we heard I would say, an impassioned discussion of the power of the IACC from Francis Collins, particularly with relation to the COVID research agenda that we've just been talking about, and pointing out the need for understanding variability of infection, risk determinants, resilience, and a number of other opportunities, possibly even in clinical trials. Followed by that, we had a presentation and I would say, a very good embrace of the IACC charter led by Mary Da Silva. I think it's worth pointing out that this remains a living document. Comments are desired from anybody at any time, and we'll continue to tune the charter to meet the overarching goals and science strategy of the IACC. Laura Rodriguez presented a breadth of work on key policies around data sharing, publications, and industry. And through the polls, we learned that these are in really good shape. I think there were a number of individuals that wanted to see targeted changes, which we have asked for the specifics of those so that we can incorporate them into each of the documents over the next couple of weeks. And then finally, we closed on a very stimulating and rapid-moving, very stimulating session on the state of COVID research from the perspective of both the genomics of the host, the aspects of sex differences, as well as mental health, and learned as an exemplar of the amazing opportunity to work within populations and maybe across populations to marshal studies that could actually help us understand both the host response to the virus as well as how the virus responds to the host as well. As I said, we're going to look for all of your ideas to help drive a research agenda around this topic. But of course, the IACC is broader than COVID-19, so we're not going to, as you'll see in the following sessions, we're not going to leave just with COVID research ideas. We're going to be asking you for your ideas on a number of other research topics, several of which have come up recently and that have nothing to do with infectious disease.